particular section, we will now look at physical controls that are used to control microbial growth. So the first one we want to look at is heat. You know, when you visit any kind of grocery store, you see that there are many canned goods that are present all over the store, and there's lots of food that is all over. The best way to really sterilize food is to sterilize it in a laboratory media or glassware or any kind of equipment. What happens is that heat will kill the microorganism by a process which is called as denaturation. Some of you may remember this term denaturation, which means that the three-dimensional shape of the proteins are destroyed. So here is this protein in an active form, which is folded. When you heat it, then the temperature causes certain hydrogen bonds, certain sulfur bonds, covalent linkages, and henceforth to break, and you have this unorganized chain of amino acids or polypeptide, and that's called as denaturation. Boiling will do that because you have high temperature. Steaming definitely will do that, again because steam has very high temperature. When you place an object or an organism or any kind of instrument in an autoclave that under steam and under pressure, that would do that. Pasteurization means that you provide heat for a short duration. That will also do that, which is used for milk. Dry heat, such as flaming, that we will do a lot in the labs, in which we will do flaming of the loop. So pretty much there's an inverse relationship between heat and time. If you heat it at a very high temperature, then you need very short time. If you do not have that high of a temperature, then you have to increase the duration. So that's what an inverse relationship means. Filtration is another method of separating out microbial growth. The filters that are used they have pores that can almost take out usually even 1 micron, in length, and they will be able to do that. Filtration is used, and for example, in the filtration plants where we have our water filters and stuff like that, that's what they're using. Their chambers that will have very tiny pores, and they will trap the bacterial cells. And cold, which is called as bacteriostatic, which means that extreme cold temperature will also kill them. And that's why sometimes when you place something in a freezer at a very, very low temperature, that will also reduce their activity, because the microbes do not produce that much toxins, and they will not destroy the food. However, there are other kinds of microbes. For example, the psychrophobes, which, if you remember, those were the ones, the psychotrophs, or the psychrophiles, that will grow in the cold environment if it's left for a very long time, and it will destroy the taste of the food, and it will actually make you sick if you have something that's in the freezer for a very, very long time. Some other physical methods that can also use for control growth is desiccation, which means total removal of water, dehydration, and this can sometimes, endospores are especially tolerant to desiccation. So desiccation means that you pretty much have, for example, when you have dried banana and dried peaches, those are all desiccated because the water has been removed. Change in osmotic pressure can also affect control growth. For example, salting and making pickles, in which you change the osmotic pressure, and that will affect the growth. And lastly, radiation. Radiation can be ionizing, for example, X-ray, gamma rays, or electron beams, or non-ionizing, which is the UV rays. Microwaves do not kill by their rays; they kill because of heat, and it's not really very antimicrobial. You don't really kill anything through microwave length or microwave rays. What physical method can control growth? The non-ionizing forms, which is the UV radiation is the one that will control forming what is called as the thiamine dimers. Thiamine dimers are means that there are two thiamine molecules and they are attached together to form a dimer, and once these dimers are caused, examples of thiamine based lesions are called by UV radiation. So when these are formed, they will affect growth of the organism. And sometimes these thiamine dimers are formed even during cancer cells. So that's another topic totally, but that's what a non-ionizing form will do. Another physical method that can control growth is called as surfactants. Surfactants are detergents, for example, soap, and whether it's liquid or solid soaps. They destroy the membrane structure and the membrane permeability, and they can then they can pass through the membranes, and they can destroy the microbial growth. And here is another picture of the filtration. Filtration is when you have pore sizes that are so tiny that it can trap the microbes on the top part. So here's a filter, and if you add filter to it, you've trapped all your microbes that are of different widths. So filters can range from different pore sizes. Now filters also have to be replaced at a certain amount of time. So that's why, for example, if you have a refrigerator that gives you clean, cool water, you do need to go and check its filter, because you can't leave a filter on for like years and years and years and expect to get clean water. So this was the physical means, and I'm going to stop here, and then we will look at the chemical control in our next mini lecture.